the last 2 days, the last 2 lectures, on just getting all of us warmed up because of all our different backgrounds on some basic language that will help us in understanding some of the things that we're going to move towards. And the goal for today is to now get into some more specifics on getting a sense of numbers, getting a sense of numbers in biological contexts. And what that means is getting a sense of relevant length scales, time scales, and energy scales that are important for different biological processes. So I'm going to try and make it as interactive as possible. You'll do some, try to work some problems in the class using the whiteboard. Well, you can do it. You can either use the whiteboard or you can do it on a pen and paper, take a picture and share it. Okay, so we're going to use the E. coli bacterium. So the E. coli is a bacteria, and we're going to use that as our standard ruler. So you know how, like in physics, the hydrogen atom is like the thing that you study from day one, and you try to understand all the quantum properties of the hydrogen atom, right? In biology, there are certain proteins like myoglobin. Myoglobin is like the hydrogen atom of physics. So it's one of those systems that one, has been studied for a long time to understand some of the basic working principles in biology. What we're going to do here is we're going to use this the E. coli cell, a single E. coli cell, a bacterium, as our benchmark, as our ruler. And so we want to get a sense and a feeling of so the image that I'm showing you here is an electron microscope image of a single bacterium, and we want to get a feeling of things like how much does the bacterium weigh? What's the weight of a bacterium? If you were able to take a measuring tape and dissect all these various things that you see here, what are the length scales? What is this length scale here? What is the length scale of the pili? What's the length scale of all the different entities that make up the cell? So all those things in that organic soup that we were discussing. And of course, finally, we want to get a sense of what are the time scales associated with processes in images like the ones that you're seeing. So there's a tendency, and this is something that we want to try and avoid, is to look at images in biology and think of them as static objects, but they're far from that. And in fact, for the longest time in biology, biologists used to think of biomolecules and biosystems as static objects, because they, when they solved X-ray crystallography structures, they said, look, this is the structure of the protein. This is the thing that performs its function. And it was only after theoretical models, in particular things like molecular dynamics, were done, when there was a shift in the thinking on what actually underlies the fluctuations of these things. And so these are actually breathing dynamic objects, and these fluctuations are in fact very important for function. Okay, So just to make it a bit more concrete, so this is a zoom in of our bacteria, okay, and Homer here is basically a protein, okay, or a molecule that has to diffuse along this surface. So it has to move from one end here to another. How long does it take Homer to do it? Okay, that's the type of what are the factors that control how the molecule is going to diffuse? Is it going to be a Brownian random walk? If it is a Brownian random walk, is that consistent with what we know of the timescales of biological function? What is the role of the medium? So, remember, all these proteins and molecules are diffusing through a medium that's very crowded. How do we understand these length scales and time scales in terms of all this complexity that's going on? Yeah, so that's what we're going to try and head towards answering and thinking about these types of questions. Okay, so that's looking at this from a bird's eye view. Okay, so we're looking at the whole E. coli. But of course, in the first lecture or the second lecture, we zoomed in and we now appreciate that, of course, all these biological organic entities are made up of building blocks. They're made up of lots of different types of proteins, lots of different shapes, sizes, topography, with lots of different functions. So if I was to imagine taking a protein, well, there's a lot of heterogeneity in them, but let's take a typical protein like myoglobin. What is, if I treated it like a sphere, what would be the typical radius of that protein? What would be its mass? And similarly, what would be the size of the specific building blocks, the amino acids that make up these proteins? Now, why it's really important to have these intuitions in your head is because when you start building theoretical models for biological systems, whether you do it analytically or you do it numerically, it's important to know what are the boundary conditions, let's say the chemical boundary conditions that, so, in one case, maybe it's okay to consider, to take the spherical cow approximation, which is, you make a protein a sphere. But there may be other cases where that approximation doesn't hold because it's simply not true. And so having a sense of these things will allow you to keep these things in mind as you start to think about, especially those of you who will work on more biophysics related projects. Okay. And so encapsulating all these questions is fluctuations. And of course, the thing that drives molecular processes are forces, they're energies, right? And in the cell, we have let's say a protein coupled to a heat bath, and there are thermal fluctuations going on. And so there are thermal fluctuations going on at a specific length scale. And those thermal fluctuations going on at a specific length scale drive the conversion of energy from many different forms to another. Chemical energy to mechanical energy, mechanical to electrostatic energy, electronic energy to mechanical energy. And what we want to get a sense of is how these forces are able, thermal forces are able to do this in biological systems. Okay, so that's where these are the questions. This is where we're headed. Okay. All right. So here are some of the first answers. So of course, this is one should think of these as typical numbers. There's going to be a spread of them, but the typical volumes and masses that you need to be thinking about for cells are on the order of, micrometer and picogram. Okay, so pico 10 to the minus 12 grams. The typical, let's say you were to take the spherical cow approximation and you asked yourself, what is the typical volume or mass size of a protein? Let's say the typical radius would be about 20 angstroms. But of course there'll be a variation in that. And finally, what are the energy scales that or the magnitude, the forces that we're looking at? Well, you've got kBt, that's fixed by the temperature, so thermal energy. And as you will see in more concretely towards the end of this lecture, so force is energy over a length, right? And so the typical forces that one is dealing with in biological systems is on the order of piconewtons. Okay. Now, I want to share an anecdote of something that's related to this, because when I was I remember when I was a graduate student, and I remember doing one of my first simulations of a biological system. I think it was myoglobin, and I remember using a code and something wasn't working right. And I remember going to my supervisor and saying, yeah, I'm seeing forces on this amino acid side chain that's on the order of newtons. And he was like, newtons? How did you? And I was convinced that there were these very strong electrostatic forces that were generating Newton magnitude forces. And so, well, it took me a while to eventually realize that this was not possible and didn't make sense. And so this is why this is one of the reasons why I think having a really firm footing on what you expect from data that you're generating and how it relates to different physical systems is really, really, really important, because it helps you sort of put a threshold on when a model is correct, when it breaks down, when there's an error, when there's, okay. But so we're going to come back to this piconewton story because there's this magnitude of piconewtons at the nanometer length scale makes biology very, very, very special. Okay, and this is what you're going to see hopefully towards the end of this this lecture. Okay, so now we're going to do a little exercise. So we're going to start by asking. We're going to take this E. coli cell, and we want to know how many proteins there are in the cell. So we want to make a guess or an estimate of what that is. So the cell has it's 1 picogram, and 70% of it is water. Okay, the rest of the 30% is non-water, so it's dry weight. Out of that 30%, half of that is protein. Okay, so you know what the total cell mass is. Let's assume that your average protein has 300 amino acids, and so this means that the total mass of this protein is 30,000 Daltons. Daltons is just a unit that is used it's like SI units. It is SI units for mass, and it's used often in biology because of dealing with small weights. So 1 Dalton is 10 to the minus 1.6 times 10 to the minus 24 grams. Okay, so the question is, how many proteins are there in a cell? So, okay, we're going to, you might take a guess, or I would prefer if you actually worked it out. So let's get a whiteboard. So you can use this, or you can even put it in the chat as you wish.
Ali, can you project the screen again?
Sure.
I forgot the numbers.
No. I will. Just give me a second. I just need to put this in the chat, and then I will do that. Okay. Here we go. Now I think I'm going to try and log on from another device. Let's see. Oops, sorry, give me a second. Oops. If there's someone, if you want, if there's anyone who wants a clarification of the question again, please don't be shy, just ask.
Yes, please.
So I can't, who is this again?
It's Maha.
Maha, sorry. Okay, yeah. So, okay. So we have a cell, okay? A bacterium. The bacterium is 1 picogram, the whole weight. 70% of that is water. 70% of 1 picogram is water. 30% of it is non-water, dry weight. Half of that, so 15% of the cell weight is the weight of the total weight of the protein. Okay? Okay. So now, if you know the total weight of the protein and you know the weight of a single protein, so I'm telling you that a single protein is roughly 30,000 Daltons, where 1 Dalton is this horrible number, 1.6 times 10 to the minus 24 grams. Okay, so that's okay. So I want you to give me an order of magnitude. So let's see what we have here. Okay. It's a very simple calculation, not trick here. I'm not trying to trick it. It's just take just take a ratio of two things. That's all. So Rolando, I see you've done some stuff. So what's your estimate of the number of proteins in the cell?
I'm not sure. It's that I think I'm doing something wrong because it's gonna be like
I
really...
I think you're making it. Yeah, I think you're making it a bit too complicated.
Yeah.
So
Okay.
so yeah, I mean, so you know one picogram is the weight of the cell, right? So 15% of 1 picogram is the total weight of the protein. And then you just have to the weight, the average weight of a single protein is just 30,000 times 1.6 times 10 minus 24 grams. That's just the weight of a single protein. Because I'm telling you, well, this is an assumption, but it's a reasonable, that the total mass of a single protein is 30,000 Daltons, where 1 Dalton is on the order of 10 to the minus 24 grams. Okay, so you just have to take the ratio of that 15% of 1 picogram divided by the mass of a single protein.
Oh, okay.
Okay.
Yeah. Yeah, it makes sense. Yeah.
Okay. Julia.
Yes, I'm here.
Yeah. What's up?
Still trying.
Romina.
I don't know, Eddie has different magnitudes. I have something like 300,000 proteins.
300,000.
Yeah.
300,000. How did you
Is
get
that too much? Is 300,000 too much?
No, it sounds a bit too less.
It's too less. Yeah, Eddie has something higher. I think I miscalculated.
You have something like 10 to the power 8.
10 to the power 8.
Yeah, I found something like tens of millions.
How many? Say that again.
Tens of millions, 10 to the power 7.
Yeah. That sounds better.
Yeah, 10 to the 7. So on the order of millions sounds
correct.
Sorry, Professor.
Yes.
And I have found 0.3125 into 10 to the power 7.
Well,
0.3125 into 10 to the power 7.
Okay, perfect. So 10 to the power 6 then.
7.
Yeah, well, I have the same result.
10 to the power 7, yeah. 0.3125 into 10 to the power 7.
So 3 times 10 to the power 6, basically.
Yes.
Yeah. Okay.
Then it's like Romina's.
It's it's millions then, Romina.
Yeah. I think.
Not Yeah. Okay. Okay. So it's 3 million.
I have the same number, but 1 0 less, but probably
Okay, so
was
it's
calculated somewhere.
Okay. Very good. So Eddie, 10 to the 8. I don't know. Sounds a bit much. But okay, but I think you get the point. But now, so okay, now, does that surprise you? Let's say if you didn't do this calculation, what's the number you would have gone by? Okay, let me try to lower, right? Yeah, so I would have, yes, my first knee-jerk reaction would have been, well, but that smaller number is not well-founded. In a sense, it's founded based on, I'll tell you what it's founded by. For me, it's founded by the fact that I have been doing a lot of biomolecular simulations my whole career. And what you do is you usually see nice pictures of single proteins when people do simulations. And then, we try to make inferences of things going on in the cell, but we have to be very careful. They are 10 to the 6 proteins, or that order of magnitude. And now you can your brain can start churning on various things. What's the concentration? What does that mean in terms of spacing? That's just only the proteins. What else is there? Fats, lipids, sugars, DNA, ions. Okay, so then you begin to see that, I have to really think very carefully about what type of model I have in mind when I'm trying to understand a biological process. That doesn't mean that there's no value in doing those models or those types of simulators. There's a lot of value, but at least it allows us to calibrate the assumptions and limitations. So, okay, hold on. I'm just trying to make sure that I see everyone's stuff on the whiteboard just to see the spread of the numbers. Lian, Valerius, Paul, Julia. Oh, maybe you put it in the chat so I can look in the chat. There we go. Okay, so I think people are converging on 10 to the 6. Lian, I haven't heard from you.
Yeah. I wrote it on the whiteboard. Actually, you didn't see my whiteboard.
No, I can see it, but I don't see anything on it.
Oops.
Yeah.
I wrote it a long time before.
Oh, you did? Okay.
Yeah.
Okay, so you got something on the order of 10 to the 6.
3 million.
Okay, 3 million. Yeah, okay, perfect. Very good. Okay, so lesson number 1, the cell has 3 million, on the order of 3 million proteins. Aha, okay, that's a lot of protein. Let's move on. Now let's ask, so how many water molecules are there in a cell? So let's try and solve that problem. So 70% of it is water. And
Sorry, Professor.
yeah.
Yeah, here the 3 billion we calculated, it is 15% as what? Because
Okay.
we have said 70 is water.
Yeah, let me so remember, okay, a cell has 70% water, 30% is dry weight. Okay, so the non-water mass comes from proteins, comes from DNA, comes from fats, comes from sugars, comes from ions, comes from other things. And So out of that 30%, half of that is protein.
Okay.
Okay.
Okay, get it.
Okay. So next task. Tell me the number of water molecules. And well, of course, the hint is that it must be more than 10 to the 6. So if you get a number less than 10 to the 6, there's a problem.
What is the weight of a water?
The molar mass is 18 grams per mole.
Okay.
Okay. So let me put that in the chat. Molar mass of a single water molecule, 18 grams per mole. So yeah, you will definitely need that. Sunday, this is the answer to the number of proteins, right? Yeah, this is the, okay. Just wanted to make sure. Okay, Lian, I like your number. Let me look at the whiteboard. Julia, I haven't heard from you.
For the mass of the number of water molecules or the previous one?
No, well, both. Is it?
Yes, I wrote it in the chat.
Okay, no, but you understand there's no problem. It's clear.
Yes.
Okay, good. Okay, good. Okay, Leanne, now I can see your work. Maha?
Yes.
What's
Can you see the first one?
I can see your first one right now, yeah, on the whiteboard.
Yeah, I'm still thinking.
You're still thinking, okay, no problem.
Yeah, what's one mole equal to?
Oh, sorry?
The mass of water is given here in per mole?
Yeah, per mole, yes. And well, yeah, 1 mole is Avogadro's 6 times 10 to the power 23. Okay, let me see. Let's see what there is in the chat. How do I get this? Eddie, what did you get?
I'm still doing it.
Oh, you're still, okay. just going to get a glass of water. I'll be back. Pierre, what do you get? Sorry, I'm muted.
Yeah, I got 10 to the 22.
10 to the 22. Mamma mia.
Yeah, there's something wrong.
Yeah, that's like yeah. I mean, how many atoms are there in the universe now?
Okay, so the
yeah,
question was the number of water molecules, right?
exactly.
Okay.
Exactly.
I get 20,
I
10
get-
to the 13.
How many?
10 to the 13.
10 to the 13. So so just walk me through how you got it.
I mean, I took the weight of the cell 70%.
Yeah, so 70% of 1 picogram.
Yeah, and then the weight of 1 water in grams.
Okay.
Which is like the 18 gram per mole divided
Perfect.
by the Avogadro's number.
So, and you get 10 to the 13.
I probably miscalculated it.
Okay.
Again.
Okay,
I
so
got something 10 to the 9.
Yeah, 10 to the 9, 10 to the 10 is more like what?
I have something like 10 to the 8.
10 to the 8th.
Yes.
This is it sounds a bit
finally.
And Pierre, what do you get?
yeah, I forgot the pico in front of gram, so I found 10 to the 10.
Okay, that perfect.
Yeah.
Okay, good. So 10 to the 9, 10 to the 10, that's good. That's reasonable. That you know, like 3-4 orders of magnitude larger than the number of proteins. Okay, that makes sense.
So my answer is in the chat.
So your answer is in the chat. Let me look at the chat. Hold on. Yeah, perfect. So it's Paul, right? Paul, wait.
Yes.
Well, it looks. Paul, your answer looks a bit too. I prefer the. I like the answer by Maha and the second answer by Pooja. So
yeah, the first
think,
answer is actually wrong. So
yeah, I think so. It's probably just a simple.
Maybe
missing,
simple.
It's a missing factor somewhere.
Again.
Okay. Okay. Good. So 10 to the 6 proteins, 10 to the 10 water molecules. Okay. Now, you will. That's a lot of water. Okay. Now, you will get a much bigger appreciation of why this is so important to think about. So imagine the cell cycle. Imagine taking one cell and building 2 new cells, daughter cells, from that one cell. Imagine the time scales of cell division. So what you're going to see very soon is that the time scales of generation of proteins, 10 to the 6 proteins, in order to generate a new cell, the time scale of needing to suck up 10 to the 10 water molecules on a certain time scale implies some very, very, very directed motions in the cell. You know, you would not be able to do these types of fast processes with just Brownian random diffusion. Okay, so I'm going to get there, but first we're just getting a sense of these numbers in the cell. Okay, we could do the same exercise for the ions, but it's the same thing. I'm not going to let you do it now because I think you get basically the point. So the concentration of ions in the cell is roughly on the order of millimolar. Okay, so again, you can do the calculation, and if you just focus on one ion, one positively charged ion, one species, the numbers you'll get are roughly between the number of proteins and the number of water. So 10 to the 7, 10 to the 8th. Okay, so all of this, these 3 components that we just talked about in a 1 micrometer cubed volume. Okay, so now this is where the interesting physics questions begin to start to pour in, right? And when we have this sense of appreciation of this, of how complex this problem is in terms of these different properties. Okay, so what are the implications? So let me go here, share my screen. Okay, so the take-home message here is, without considering all the other species in the cell is a very, very crowded environment. And so this is a sort of a classic textbook picture that you'll find. I forget who was the first person to author it, but it was one of these, the figure on the left here was one of these cartoon depictions that were made, I think 15 to 20 years ago, trying to illustrate and to point out to theoreticians and modelers that, you know, biology and biological systems are much, much more complicated than we like to imagine they are with the simple models that we use. And so the take-home picture is the one on the right here. I have lipids, I have proteins, I have DNA, I have ribosomes. These are the things that generate proteins. I have messenger RNA, water molecules, membrane proteins, ions, all this in this 1 micrometer cube space. Okay, now there's some, you know, we can begin to ask, what are, if I have such a crowded environment, what are the typical distances between one protein and another protein in a crowded environment? Okay, that of course is determined by the concentration of these proteins. Okay, and so I for the rest of the discussion, I'm going to ignore all other species, but just focus on proteins, because these are sort of the essential building blocks, and we'll be revisiting proteins again and again more frequently than things like DNA and ribosomes. So here's a way to see what this means in terms of concentrations. So this is a plot on the log-log scale of the concentration in nanomolar. So this is the typical unit of molar is concentration, nanomolar concentration. This is a function of concentration and the number of molecules in a cell. Okay, so the rule of thumb is that 2 nanomolar implies 1 molecule per cell. The molecule in this case for our discussion can be the protein, okay? But it could be something else in general. And so of course, as you increase the concentration, so going up to 500 nanomolar corresponds to roughly 100 molecules in the cell. If we have 10 to the 6 molecules, so 10 to the 6 proteins, that's the number that we came up with. So that's somewhere up here on this map. Then we see that you can build basically you can take the cell and you can put it on a lattice and assume that they are equally spaced, okay, just for simplicity, and you can make an estimate of how the spacing between the points on the lattice, i.e. the proteins, changes as you increase the number of lattice points, so where these are the proteins. And so the typical spacing between 2 proteins at concentrations of 10 to the 6 nanomolar basically is on the order of 100 angstroms, so 10 nanometers. So in a cell, a protein is basically seeing another protein about 100 angstroms away. Okay, that's so 100 angstroms it's not so large. And it's not so large because remember, besides the protein, there's also the liquid around the protein that gets perturbed. So, okay, let's imagine that the liquid around the protein is perturbed by the order of one nanometer. It's actually between 1 to 2 nanometers, but let's be conservative, 1 nanometer. The other protein also is perturbed by 1 nanometer. So essentially now we have 80 angstroms of bulk-like liquid that separates 2 proteins. Aha, but there's not just proteins, right? There's ions, there's other stuff there. So the picture gets much more complicated as you start to think about all these various nuances that are there in the cell, but often very difficult for us to capture in a simple model. Okay. 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 So I think, well, basically, this is something I already described. I already said in words. But the take-home message here is we have a very crowded environment, and there are some very interesting questions about the nature of the liquid in terms of its viscosity, the effective friction. If you start thinking about, a diffusion process of the protein through the cell, the cellular environment. Okay, are there questions before I move on? Okay. Well, okay, so, and this is just one last, maybe more practical comment. We often want to be able to compare with experiments, and any type of, that's where usually biophysics usually happens first with an experimental measurement of something. And if there's only one thing I want you to take away from all our discussion today, is that, you have to be very careful with the difference between an in vitro and an in vivo experiment. In vitro experiments are typically how experiments are done, biological experiments. They're done in a lab, in a test tube or a Petri dish. And what you learn from that is useful and interesting, but it's not quite the same as what is happening in the cell. And that's because the environments are much more complicated. And this can change the physics as well of what happens. And this has been shown, in fact, in several experiments. Okay. So we've now covered a sense of numbers when it comes to numbers of different species. But as I pointed to you before, these are not static, right? These are dynamical things. So of course, when we say there are 10 to the 10 or 10 to the 6 proteins, that's this thing is fluctuating, it's breathing. These are dynamically changing things. And so we want to now get a sense of the important relevant time scales that one has to bear in mind when we start to think about biological processes. And I think this is actually let me say that one of I would say the big grand challenges of modeling or of theory in general in biophysics, soft matter biophysics, is it's because you're dealing with soft matter systems. There are fluctuations unlike in the solid state. There are a wide variety of timescales that one has to consider, and this makes it very, very challenging. Okay, so let's build up on the timescales that are relevant. So let's take our molecules. Well, I'm sure all of you now, you all took a course in molecular dynamics, either at CISA or you're doing it now at ISTP. And one of the things that you for sure discussed was choosing your time step in your molecular dynamics integrator, right? And at the heart of that question is the timescales of the relevant processes, or the time step that one has to choose in most biological systems. The limiting step is high frequency modes, right? So if you take a water molecule, the water molecule has covalent bond vibrations. Proteins as well, they're made up of covalent bonds, and these covalent bonds vibrate on the femtosecond time scale. So that's I would say the fastest time scale of the nuclear degrees of freedom in a biological process. And they happen to be very relevant as well for biological function. When you start to think about processes involving electronic excitation, so photo excitation. Okay, so that's sort of the fast limit of a fast time scale that one has to bear in mind in processes in biology. Then the next is picosecond. Okay, so the typical picoseconds are the time scale of hydrogen bond breaking and forming of water. Okay, so that's sort of say not the rule of thumb, but that's the typical. And all these time scales, of course, are related to a rate theory, right? So the time scale comes from an activation barrier. There's an activation barrier associated with all these various processes, so bond breaking and bond forming. And so the rate of picoseconds comes from the size of that, the relevant barrier associated with the process. Okay. Yes, Leah, go ahead, please, go ahead.
For this parameter, when we do the modeling, we will take it from the experiment, or we will calculate it and then.
You mean like the time scale of picosecond?
Yeah.
Okay, so well, so remember, okay, you remember ergodicity, right? Yeah. Okay, so let's say if I do my model or my simulation on the nanosecond, on the long nanosecond time scale, then I'm safe to say that I have essentially explored the picosecond timescale processes correctly. So no problem there. So it's not something that you really put into the model from that perspective. Now, on the other side, if there are processes with very large activation barriers, then your simulation on the timescale of, let's say, nanoseconds will not allow you to hop over the barriers. And so then you have to do some other smart things to get the rates and to get activation barriers. And this is stuff that the PhD students from CISA are doing right now with Alessandro's course on rare events and things like this. But yeah.
Yeah, I'm a bit curious. How can we track the where we have the bond breaking, for example, or bond formation, or when we have the intermediates or something like that in our simulation? Like when we track it. Okay, how can we know at that time and in how long, so that we have the bond breaking or formation where we have the intermediate, like we based on the energy or something?
Yeah, exactly. Yeah, that's exactly it. Yeah, there's a knowing the energy scale of your different interaction relative to thermal energy, you will be able to fix the time scale of the process. So I'm going to get to your question shortly, more specifically. So if you just hold on for like 5 minutes, and then I'll get to your question.
Ali, can I ask a question?
Yeah, please go ahead, Eddie.
I'm curious about this this time, like when we choose the time step for a molecular dynamics calculation.
Yeah,
we look at the frequency of the vibrations. But what if we are looking at a quantity which does not rely on vibrations? For example, like heat capacity of water, for instance, where I mean you just model the molecule as like a rigid state, you know?
Yes.
So, and it reproduces the heat capacity properly. So in that case, how would you choose a time step?
Okay. This is a very good question, and so let me see. So okay, so different processes have different time scales. Okay, now this is not a very profound statement, but let me try and clarify what I mean. So okay, if you took a covalent bond, like an OH bond, and you were to measure its decorrelation time, Okay, so the time scales that over which it would be fluctuating at equilibrium, that would be femtosecond. Okay, but now let's say you were to look at the energy. So the energy which goes into the heat capacity of the system. That may be relaxing on a slower time scale. Okay, and so that what that means is that, you know, so there are two things. One is I need to first, it's not okay. The fast time scale dictates how I choose that time step that takes the fastest modes in my system. And if I was interested, let's say I was interested in spectroscopy. So, okay, so you gave the example of heat capacity where having the water rigid or not may not make such a big difference. But let's say I was interested in a type of spectroscopy where the electrical charges of the water molecule fluctuate and reorganize. So like in infrared spectroscopy. Okay, so in infrared spectroscopy, you have a transition dipole that goes into the selection rules through Fermi's golden rule. And if I don't have the actual bond vibrations, and I don't integrate through those fast timescales correctly, then for a measurement like spectroscopy, that would be problematic. So it depends on what experimental observable one is looking at. So yes, I mean, heat capacity is less controlled by when you're in the classical limit. So at high temperatures, you don't need to worry so much about the bond vibrations. Okay, so there are other heavier and slower degrees of freedom that dictate the heat capacity fluctuations. But that's it depends on the process.
Yeah. So I guess essentially, if the process does not depend on vibrations, then you have like a high degree of freedom to choose what your time step is.
Exactly. Yeah. Exactly. And but by vibrations, so vibrations mean different things to different people. So like localized high frequency vibrations.
Okay.
Yeah. So if the process you're interested in is sort of separated from that, the physical process, then you can safely get away without doing it. Yeah.
Thanks.
You're welcome. Okay. Yes. So okay. So we have picosecond, and then now let's imagine our 10 to the 6 proteins in the cell. Of course, they're not static. They're going to be breathing entities, and they can tumble in solution, right? So they'll be tumbling because they'll be, being punched from different sides by all these liquid molecules that surround them. And of course, depending on the size of the protein, the tumbling time, so how long it would take to rotate in solution, will change. So for a small protein, by small I mean I don't know, 30, 40 amino acids, typical time scale of tumbling is on the order of nanoseconds. Okay. Okay. Then there are processes that that go beyond, so microsecond, millisecond, and these are things that are then become much more relevant to, let's say, really biological processes, so like a protein folding. So the protein folding problem, and we will discuss this in detail much later in the course, is the problem of taking a polymer it's like a spaghetti chain, and how it becomes a collapsed folded structure. So that whole process of protein folding can take place between microseconds to milliseconds. Okay. And similarly, if one of the let's say the holy grails of biological processes in the cell are interactions, right? I have different proteins, I have different molecules, and in order for them to do their function, the things need to stick to each other. When you take drugs, medicines, they function because those drugs go into your bloodstream and they stick to specific sites. How that sticking takes place and the timescales of how that sticking takes place, occurs typically on the millisecond timescale. Okay. Okay. So this basically just summarizes the whole story.
May I ask?
Yeah, please go ahead, Leanne.
Can you go back to the
go back.
slide?
Yeah, this one.
No, this one.
Next one. Okay.
Yeah. I'm wondering which is the x-axis in y-axis we have energy, right? But in x-axis
the
I don't know.
Yeah.
What is that?
Okay, I glossed through this because I didn't want to do it because it's actually wrong. Anyway, this is the we're going to come to this later on, but this is basically, it's called it's the energy landscape of protein folding. So on the y-axis is energy, and on the x-axis is an order parameter that allows you to say the polymer is folded versus unfolded. Okay, so it's just some generic order parameter that says which is exactly on this axis too. So it's like for example, the radius of gyration of a protein, of a polymer, or the number of contacts. It's just a measure of how folded or unfolded the protein is. But don't take this thing literally, because it's a picture that the right one that's used a lot in the protein folding community, but it's actually wrong. But anyway, we're going to come back to that later on. Okay.
Yeah. In that we should expect that the lowest energy should be in for example folding. So the radius should be small.
That's okay. So yeah, that's so basically that's the thinking is that that the lowest, if a polymer has a stable folded structure, then its native structure, or its thermodynamic, its lowest free energy structure will be something which is folded. But yeah, the problem with these pictures here is that the way people use them is they often sometimes think of the y-axis as potential energy, and this is very wrong, because the relevant object is the free energy, and so you have to think of both contributions of entropy and enthalpy in this. Yeah. Yeah. Okay, so this is the big picture. Just to give you a sense of, okay, I'm not going to go through this, but you'll see that you go from 10 to the minus 15 femtoseconds. That sets the time scale of the fastest motions. This is the region where interesting, where quantum mechanics, where electronic structure plays a role in biological processes. Okay, so for example, vision. When you see things, this is because there are specific proteins in your retina that are absorbing light at the right frequencies, getting electronically excited, and triggering off a whole cascade of processes. So converting that energy, electronic energy, into mechanical energy that then propagates to the system. Okay, but this is a very interesting time scale and region to work in, because this is where there's a lot of interesting open challenges and questions with the role of quantum mechanics in biological systems down here. Right? Then as you move, you cover a whole range of timescales up to nanosecond, microsecond, millisecond. We already mentioned some of these things. And of course, then there are, processes that take place on the timescale of seconds. For example, the generation of cells, right? So cell division takes on the time minutes, actually. Okay, so take-home message: broad hierarchy of timescales. Ergo, this is a challenge, and this is why it's this broad hierarchy of timescales which has motivated essentially all the interesting things in advanced sampling techniques, sampling problems. This is, in the context of biological systems, is rooted from the fact that we have this challenge of, covering all these large timescales, and we cannot do it just by brute force. Okay, so we have about 5-10 minutes. I want to just sort of try to conclude this part with putting into perspective now and connecting the numbers I showed you on the number of proteins and etc. and the timescales. So there's a process known as cell division. You take one cell and you generate 2 cells from it. It's a horribly complicated process. Don't ask me how it happens. I don't understand it myself. There's a lot of complex biochemistry. So I can't explain it to you, but all I want to discuss is how to relate it to the things that we're interested in discussing here. So I take one cell up here and I generate 2 cells. Cell division. This takes about 3,000 seconds. This whole process. Okay. Now let's think about this. So I have to take one cell with 10 to the 6 proteins, 10 to the 10 water molecules, 10 to the 7 ions, and I have to do some work to generate 2 of these objects with the same number of species. Okay, so so essentially, well, you all know that the essential building block of proteins or of organic matter is carbon, right? So we're mostly carbon-based species, and so you can think of this as like the construction material. And so in order to generate new amino acids, the cell needs glucose. Okay, glucose is a sugar, and using the numbers that we had before of 10 to the 6 proteins, roughly you need about 10 to the 10 carbon atoms to generate a new cell in the cell division process. Okay. Now let's do the math. So if you need 10 to the 10 carbon atoms to generate a new cell, and the cell cycle takes 3,000 seconds, a glucose molecule has 6 carbon atoms. Okay, basically you need to absorb 10 to the 5 glucose molecules per second, okay, in the cell division process. So that's quite efficient, right? So that's number one. Number two, when I generate a new cell, I need to produce 10 to the 6 proteins, and I need to suck up from my water reservoir 10 to the 10 water molecules. So in order to do this, I need to generate 1,000 proteins per second, and I need to absorb 10 to the 6 water molecules per second. Okay, this is it's quite humbling, right? I mean, it's quite an efficient task that biological systems have evolved over to achieve these this type of efficiency in how proteins are generated, and we haven't even got to what is the underlying chemistry and stuff that underlies, for example, the generation of proteins. But you know that proteins are made up of amino acids. They're covalent bonds that link them together. So there must be some very efficient catalysis and directed interactions that are driving this type of efficiency in the generation of a new cell. Okay, so I alluded to this thing about directed motion, directed diffusion, and so I want to sort of conclude my lecture with this point. So because it's related to how, if there are these very fast processes, efficient processes happening on these short time scales of many particles, would we expect that if the particles were undergoing Brownian random walk, Brownian motion? Okay, so and sort of just to anticipate the answer is no. So if you just had passive diffusion, basically we would all be dead, and probably evolution would not, nature would not have been able to evolve and design, create new machineries that work optimally. So let's make a very simple argument of this. So if a particle was undergoing random diffusion, then the time scale that it would take to move a certain distance L or X would go as X squared over the diffusion constant. Okay, that's just the definition of diffusion, the diffusion constant. So now let's go back to our bacteria cell, and let's assume that I have a protein that's undergoing a purely diffusive motion in the cell, and let's say that it wants to cross the length of the protein, which is about 1 micrometer long. It wants to diffuse across the length of the cell. So let me here. So let's imagine the protein was marching along via a Brownian random walk along the cell. So you can estimate the time it would take to do that by what we know the length. We know that we can estimate the diffusion constant. And the way you estimate the diffusion constant is by using something known as Stokes Einstein. So Stokes Einstein is a hydrodynamic theory, one of the simplest hydrodynamic theories that allows you to relate the diffusion constant of a particle to thermal energy, the size, and the viscosity of the medium in which it's moving. And so by taking a simple, let's say again, a spherical cow, a protein with a radius of 25 angstroms, using the bulk viscosity of water, the diffusion constant is about 100 micrometers squared per second. So it would take 1 millisecond for the protein to diffuse across the length of 1 micrometer. Okay, so is that slow or fast? How do we, it's not clear actually. Should I be surprised by this number, or should I be concerned, or should I think it's okay? Okay, so in order to, let's say, to build some perspective on whether this number is small or fast, let's think of a specific biological process. For example, response to heat or stimuli, and so in the body there are these neuron cells. These neuron cells are one of the longest cells in the body, and response to stimuli involves the marching of proteins along this tube-like object called the axon. Okay, so whenever your body responds to something and there's some signals that are propagated through the neuron cells, that involves some, mechanical response and also chemical response of proteins in response to that stimuli. So in this neuron cell, the length of this axon is about 10 centimeters. Now, if I was to ask the question, how long would it take for this protein to diffuse along this 10 centimeter axon tube with a time scale of 0.01 seconds, you get a number like 3 years. So the moral of the story is that if proteins were diffusing via Brownian random walks in the cell in response to biological processes, basically we would all be dead. And so there must be something else going on. And that something else that's going on is directed motions that are dictated by specific interactions, by specific external potentials, by osmotic pressures, by chemical potentials, by all these external forces and directed interactions that dictate, that in some sense reduce the dimensionality in which the system can explore. Okay, so I think, well, we're out of time, and so we'll stop for today. And so Liana, well, I promise you from the next lecture we will be able to answer your question. Okay, so in the next class we're going to go to we've covered length, time, and move now to energy scales.
So just a quick one.
Yeah, please.
Yeah, so should we assume that there is nothing completely about Brownian motion, or they are there and they are
yeah, okay, for sure. There are biological processes where Brownian motion will occur. For sure, there are water molecules diffusing around a biomolecule are, unless there's a very strong external potential that leads to traps in the system, Brownian diffusion would work fine. So yeah, so thank you for asking that. Yeah, my message is not to say that everything is that there's no Brownian motion going on. There is, but there are very important directed and specific interactions when you get to particular length scales that dictate that cannot be well characterized by a standard Brownian diffusion picture. Okay.
Okay.
Any other questions? Okay, something in the chat. No, okay. Okay, very good. So I think we meet on Thursday again. And this week we also meet on Friday at 9 a.m. So same time.
Thank you.
Thanks a lot. Thanks, guys. Have a good day.
Bye-bye.
Bye-bye.
Bye.
Ciao, ciao. Ah, wait.